Okay, well, that's been a couple minutes, so I think we'll get started with the people that we have. And of course, this is being recorded, so we'll share it uh, with anybody that wasn't able to make it. So good evening, everyone. And thank you for taking the time out of your busy schedules to join us tonight. I'm Lynn Gerardin, a nutritionist with BeefSmart, and I'm your moderator for the evening. Feel free to post your questions in the chat as we go. And after Dr. McKenzie's presentation, we will have time for questions and we'll answer the questions that were posted. Or if you sent questions ahead of time to Alana, we'll make sure that she reads those out to Dr. McKenzie and he can answer them as they came up. So please do not hesitate, to turn your microphones on after the presentation, if you would rather pose your question that way instead of writing it in the chat or the Q&A. Yeah, whichever way is totally acceptable. So again, this is being recorded and will be sent out afterwards. I believe Dr. McKenzie is posting it on his YouTube channel also. So there's multiple ways of being able to see this presentation afterwards or to refer back to it. So to limit background noise, I will keep most participants muted while our presenters speak. But please, again, feel free to unmute yourself if you have a question at the end. So our presenter tonight is Dr. Derek McKenzie. He's a professor at the Faculty of Agriculture, Life and Environmental Sciences at the University of Alberta, with 25 years experience studying soil biogeochemistry. Dr. McKenzie started his research career studying forest fire as a natural disturbance and the soil impact of its long-term residue biochar. Over the last 10 years, he has moved into agriculture soil health research, studying regenerative practices and soil carbon sequestration. He is the principal investigator of the Soil Plant Relations Lab and has collaborated successfully with many ag industry partners, including farmers, First Nations, agronomists, government, and soil testing labs. His work on the impact of compost and biochar on soil health in agriculture is contributing to BMPs for regenerative practices in central Alberta. He works closely with many different communities and is always happy to share knowledge. Dr. McKenzie will discuss the importance of carbon in the soil and its relationship to soil health. He will also touch on how to define soil health, soil micro-plant continuum, and the importance of carbon in regenerative agriculture. So with that, the floor is yours.
Great. Thanks, Lynn. Thanks for that introduction. That's super nice. So I'll just share my screen and get my slides up and we'll start. Can everybody see that okay?
Yep.
Okay, so as Lynn said, so the title of my talk tonight is Soil Carbon, the Backbone of Soil Health. And my name is Derek McKenzie. I'm a soil scientist at the University of Alberta. And this is a webinar for BeefSmart Consulting Inc., March 9, 2026. And I'll just say right now that this work is has multiple contributors in this, right? So it really should say my name et al, or meaning and everyone else who's contributed. And that includes a lot of graduate students, a lot of producers, a lot of agronomists, regional research associations, and industrial partners. And I'll touch a little bit on some of those contributors in a moment, but it's really not just this presentation is the culmination of work of many people. So I just wanted to share a personal background a little bit so you guys get a feeling for who I am and what I do. So my personal background, my pronouns are he, him, but I try to respect everybody's lived reality. I'm a husband and a father. So my wife Jen, is the most intuitive people manager that I know, and I often seek advice from her for sort of graduate student management. And my son Bern is a Lego and Minecraft wizard, and I often seek advice from him on Lego and Minecraft. And my hobbies, other than soil science and agricultural soil health, are mountain and gravel biking, tree planting, Minecraft, Lego, and a trading card game called Magic the Gathering. A little bit of my background as a professor and research scientist. So I conducted my graduate studies from 1996 to 2004, where, as Lynn mentioned, my research focused on forest soils. I'm just going to grab my laser pointer here. My research focused on forest soil, silviculture, fire ecology, and biochar. And at that time, I had opportunities to connect with the forest industry. I got my position at the University of Alberta as an assistant professor in 2010 to 2017, where I continued to conduct research in forest land reclamation with biochar, and I moved sort of out of the forest sector and into oil and gas industry, and sort of was able to connect with oil and gas industrial partners and government. I became an associate professor 2017 to 2025, where my research at that point switched to agricultural fertility and biochar, soil health quantification, data science, and machine learning. And at that time, I had an opportunity to connect with many different producers, ag industry partners, digital industry, research NGOs, and government partners. And last summer, I became a full professor. So 2025 to present, I'm continuing to connect and do research in agricultural soil health. And I believe, and I hope and I believe that the future is bright here. So I'll just move on to a quick outline of my talk today. And as Lynn said, if you have, I'm going to go over quite a bit of material. And if you have questions, I would be more than happy to try to answer them at the end of the presentation. So if you want to jot them down to remember them, that's probably a good idea. So when I go to lectures, I continuously sort of write notes to myself. I'm trying to convince students today that writing notes is the best way to learn new material. And even if it's a colleague of mine that I've seen present on the same material a dozen times, I still take a bunch of notes just because it's sort of keeps me engaged as well. But so my outline tonight, I'm going to start off talking about what is soil health, how do we measure it, why do we care about it, and how do we preserve, increase it. I'll talk about the soil micro plant continuum and why that's important and why I think it's disrupted by synthetic fertilizer agriculture. We'll talk a little bit about the 6 C's of regenerative agriculture, and evidence to support carbon as a modern fertility best management practice. And that's going to be sort of the idea of carbon and synthetic fertilizer together. And I'll show you some data to support that. And then I'll talk about a new research project that I'm proposing this spring that I think will have relevance for this audience here of mixed producers. And then I'll sort of wrap up with the idea of soil carbon and how the historic and modern theoretical frameworks are different from each other, and their language is changing. And so what I'm trying to promote is the idea of a cultural shift leading to a change in practice that looks at using carbon as a required fertility product. And hopefully I can convince you of that with some of the evidence I'll show. But otherwise, we'll hopefully have a good discussion about that. Okay, so introduction. What is soil health and why do we care about it? So the U.S. Department of Agriculture says that soil health is defined as the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. Healthy soil gives us clean air and water, bountiful crops and forests, diverse wildlife, and beautiful landscapes. And so the real key point here is that are these two things. So the concept of soil to function, so soil function as a vital living ecosystem. So really more than the sum of the parts of sort of biotic and abiotic components, but a living ecosystem in and of itself. And my faculty used to have a slogan that said, without agriculture, you would be naked, hungry, homeless, and thirsty. And I really think you could say without soil, without healthy functioning soil, you would be naked, hungry, homeless, and thirsty. And so there's been a number of articles recently talking in the literature, in the science literature, talking about the concept and future prospects of soil health. And then there's a direct answer to that paper called Soil Health at a Crossroads by Philip Beebe. And so this first one by Lehman et al. Really sort of showcase how soil health, if we could add biological measurements to the typical soil quality measurements we had, and we would be getting a better perspective on soil health. And Philip Beebe wrote a response to this article saying that he didn't think that was possible because soil health is hard to define and because it's different in different sectors. And so, for example, a healthy forest soil may not be a healthy agricultural soil, it might not have the same functional capacity as a high functioning ag soil. Philip Beebe doesn't think that it's possible to quantify soil health because we're talking about different things in different sectors. And while that's true, I still think we need to move towards quantifying soil health and with that understanding that we're going to have different definitions and maybe different ways of measuring soil health in different sectors. And so This last paper that I have my students read is by Henry Anzen and all, and it's the soil health metaphor, illuminating or illusory. And in the end, he comes to the conclusion that it's illuminating. And I tend to agree with Henry that this is an illuminating concept, that the idea of soil health is not unlike human health. And so we can quantify sort of a healthy human. And a healthy human is just a human being who is able to function at a high level or an adequate level, right? And that means they're mobile, they walk around, they consume food, they digest food, um, they're healthy. And I think that it's fairly straightforward to say what is a healthy human being versus an unhealthy human being. And so I believe that we should be able to do the same thing for soil. We should be able to say what is a healthy soil or a high functioning soil versus a low functioning soil. But when we get into trying to describe, different levels of soil function, it's complicated. And it's not unlike with human health. If you could have a healthy person and an Olympic athlete who's also a healthy person but has a higher level of health theoretically, and quantifying the exact difference between that healthy human and an Olympic athlete, healthy human is tricky. So I think so. Basically, what Henry Jansen was saying was that it's a metaphor for function. It's a metaphor that the public should be able to grasp onto, given that health is a sort of a concept that we're all familiar with. And so some of the key reasons why soil health matters is one, for food security. Healthy soils yield more nutritious crops and livestock feed, forming the bedrock of food systems. Water quality and management. It absorbs and filters rainwater, preventing runoff, erosion, and filtering pollutants, ensuring cleaner groundwater and surface water. Number three, climate regulation. Soil is a massive carbon sink, storing more carbon than plants in the atmosphere together, and therefore helping mitigate climate change. Damaged soils release carbon. Biodiversity. It hosts a vast web of microbes, fungi, insects, and other organisms crucial for ecosystem function and supporting pollinators and wildlife. Resilience. Healthy soils are more resilient to extreme weather like droughts and floods, while degraded soils exacerbate these impacts. And number six is finally economic stability. Productive soils support agriculture and forestry, reducing reliance on costly inputs and protecting livelihoods. So I think there's very strong arguments for why we should care about soil health. So the next one is, how do we measure it? So currently there are soil health scoring frameworks that exist, and they're sort of being developed in the literature currently. So this is sort of cutting edge soil science today. And the first one I'll talk about is this comprehensive assessment of soil health, which was developed by Cornell University Soil Health Laboratory in the US. And it basically it measures, it quantifies different physical parameters, including water holding capacity, surface hardness, subsurface hardness, and aggregate stability. It quantifies some biological parameters, organic matter, ACE protein, soil respiration, active carbon, and then it quantifies some physical parameters, including soil pH, extractable phosphorus, extractable potassium, and minor elements. And then based on a statistical framework, it gives a score to that. And then the score is sort of regionalized. And so you can see in this soil that it's scoring very well on chemical function, but it's scoring kind of poorly to very poorly in biological function and physical function. And so the overall score is 51. And this scoring system has been developed for northeastern North America. And so the scoring functions that are used in this won't work in Alberta, but this kind of a framework can be adapted to Alberta with data. It's really a data science problem. And so this framework has been adapted to Quebec. It's been adapted to Ontario under the Soil Health Assessment and Plan Guidebook. But then there's also the Soil Health Institute in the US, which is benchmarking soil health, and they're using a slightly different statistical framework. So what they're doing is they're using very similar indicators of soil function, but instead of looking at this sort of cumulative normal distribution statistical function, they're looking at reference sites, and they're comparing sort of conventional agriculture or baseline soil function to regenerative practices or practices that promote soil health to reference sites. And the reference sites are typically grazed pasture, hayfields, and or fence lines. And so what we'll see in a moment is that the reference sites have all of the attributes for very high functioning soil. Whereas, and we can compare these as a benchmark. And so I'm tending to lead more and more towards the Soil Health Institute's model, and I'm actively trying to propose a research project that collaborates with the Soil Health Institute and the Alberta Independent Agrology Consultants to create enough data to generate a soil scoring framework for Alberta that is similar to the Soil Health Institute. And then there's a number of different labs that offer this analysis. And so A&L Canada offers a soil health analysis. Down to Earth Labs in Lethbridge is about to become a partnership lab with the Soil Health Institute and offer this soil health benchmark scoring. And I'm in conversation with Element Labs in Edmonton to sort of include this as well, but they're not as close. And so Down to Earth Labs in Lethbridge is the closest sort of commercial lab in Alberta that is going to be very quickly generating this Soil Health Institute type data. So I think that's encouraging. Introduction. So then how do we preserve it, increase it? And so here are the principles of soil health. And there's four of them, and I'll describe these briefly, and then I'll sort of talk about why I think carbon is the backbone of this, right? So the first one is minimize disturbance. So minimize tillage, minimize disturbance. The second one is increase biodiversity, both in crops and animals. Maintain a living root. So that's perennialization of a site. So moving sites into pastures or hayfields, which is obviously not possible when we're trying to grow annual crops. Um, and then maximize soil cover, which is possible when we're growing crops to try. And I'll talk a little bit later about an intercropping research project that I want to propose. But basically, if we look at another figure that's similar, what we can see is that this sort of maximize living roots and minimize and maximize biodiversity is generally feed. We can think of that as feeding soil. So this is fuel soil biology, improve resilience, and improve soil organic matter content. And then minimize disturbance and maximize soil cover is in order to protect that carbon content. So we protecting soil aggregates, organism habitat, and soil organic matter. And so really all of these factors boil down to soil carbon and what is the state of soil carbon in your soil. And really, so the rest of this presentation is going to talk about how we can increase the amount of soil carbon under best management practices and why we need to do that. Okay, so we'll sort of shift gears now and talk a little bit about the soil microplant continuum. And so just quickly, so what does soil do, or what does carbon do in soils? And so primarily, first and foremost, carbon is a source of energy and building blocks for microbes. So soil microbes eat carbon, and in eating carbon, they release nutrition. It's a source of nutrition for plants. So all the excess nutrients that the microbes don't take up is available for plants, and the carbon in the necromass from the microbes is stabilized, and we'll talk a little bit more about that later on. But then long-term accumulation of carbon increases aggregation in your soils, which increases water retention and is also very important for pH and buffering and cation exchange capacity. So some pretty important parameters for the function of carbon in soils. And so what is the soil microplant continuum, and why is it disrupted by synthetic fertilizers alone? So I show this figure. This is the nitrogen cycle, and I show this every year in my introduction to soil science class. And we'll just look at it sort of really broadly, and we'll sort of look at, so this is the organic matter pool here, and these are microbes. So these microbes convert this organic matter into biomass, and any excess ammonium they don't use is released as plant available ammonium, which is taken up by plants. So this is the soil organic matter plant, or the soil plant microbe continuum. And then when plants die, they contribute that biomass back to the soil organic matter pool. If there's any excess ammonium that plants don't take up, it gets converted to nitrate, and nitrate, because it's an anion in soils, it can be lost from the soil system. So any excess nitrogen can also be taken up by plants, but any that's not taken up can be lost to leaching through sort of percolation through the soil profile to groundwater. And if it reaches a sort of a wetland, as traveling through groundwater to a wetland, so a high carbon area, it can be denitrified as a source of oxygen-rich molecule for denitrification and basically return to greenhouse gases. And so this is the plant. So this is the plant soil microbe continuum. So we have this sort of evolutionarily adapted continuum where we have organisms, plants relying on microbes to convert organic matter to nutrients and then to take up nutrients. And when we use synthetic fertilizer, we disrupt that. So we lose this soil organic matter pool. So if we have fertilizer additions, these are already additions as inorganic nutrients, let's say ammonium and nitrate. And so we have this pool of ammonium and nitrate, which is available for plant biomass. But because we potentially have a very large pool of ammonium, it can be converted to a large pool of nitrate by nitrifiers and lost through leaching, which can lead to pollution, right? Eutrophication or denitrification as a greenhouse gas. And so really this is a disruption of this soil plant micro pool because we're removing organic matter. We're removing this organic matter cycle. So soil carbon, so synthetic fertilizer disrupts the soil microplant continuum, but it's necessary to replace mineral nutrients lost from removing biomass. I think we all know that we have to put synthetic fertilizers back because we're removing mineral nutrition in the grains that we remove annually from the plant biomass we remove annually from the site. But does removing plant biomass result in the loss of carbon too? And that's a question mark there. And I think it does, or at the very least, it doesn't increase soil carbon content over long term. And so tillage. So if we look at tillage, we know that tillage is a disruption of soil aggregates, results in soil carbon loss, increased erosion, reduced water retention. And I think we can ask this question, and I've started to do this a little bit in my lab. Has conservation tillage increased soil carbon content or stabilization in soils? And so this is a hypothetical framework for this is soil carbon content in grams per meter squared and year. And hypothesis one indicated that with tillage, we expect there to be a decrease in soil carbon content due to disruption of aggregates and increased aeration of the soil, increased decomposition rates. Hypothesis two here suggests that over time there'll be a stabilization, so we'll reach a new equilibrium of carbon content in soils with this sort of with tillage, and that in hypothesis three, if we move into a period of conservation tillage or no tillage, there should be an increase in soil organic carbon content. And I'm not sure that's true. What I think is more likely to happen is that we have a continuing decrease of soil carbon until we get to no-till, in the 80s to 2000, and this is a sort of a flat line here. And I don't think that conservation tillage or no-till by itself is enough to replace the carbon lost through cultivation over 80 years. And I have some recent data set from a recent PhD student of mine, Camila Cardosa. This is from her 2026 PhD thesis from the U of A, showed that so we looked at the soil quality monitoring project. We looked at these 42 benchmark sites across the entire province of Alberta, so one site in each eco district. And when you look at the sites that moved over that time period from high to low tillage or high to zero, there's not a statistically significant increase in organic carbon. So this is organic carbon in percent on the y-axis. You can see there's no statistical increase in high to low or high to zero or even zero to zero. There's a little bit of an increase, but it's not statistically significant. So to me, I think this says that conventional tillage by itself is not enough to increase soil carbon. And we really want to be increasing soil carbon content in order to increase soil function and soil health. And so I think that leads to a paradigm shift where we need to add carbon to our fertility best management practices. And it sort of is, I think it, brings up these 6 C's of regenerative agriculture. And so we'll start down here on this triangle, the 6 C's of regenerative agriculture. So number 1 and 2 are compaction reduction, so reduce traffic in the field, and conservation tillage or no-till. And those two are meant to protect carbon in the soil. So this is carbon protection. And then number 3 is crop and animal diversity. Number 4, continuous living plants. Number 5, cover crops. And those are really meant to be a diversification and perennialization. And then number 6 would be using compost and organic amendments as part of our fertility regime. And so these are going to represent carbon inputs. And so you can see that on this side, the top couple are maximizing carbon gains and the bottom couple are minimizing carbon losses. So those are the sort of 6 C's of regenerative agriculture. And so we set out to do some experimental work to look at C6, which is compost additions and field trials. And so this is a a collaborative research project that is, we're 4 years into this, and this is between the Soil Plant Relations Lab, which is my lab, and this project was executed by Jocelyn Kowalski, my lab coordinator, Camilla Cardosa, a PhD student, Mohammed Kodai, a master's student, Gino Karimi, a master's student, and myself in the Department of Renewable Resources, Agricultural Life and Environmental Sciences faculty, University of Alberta. And this is in collaboration with some industrial partners. So Altroot is the compost producer. So this is a compost producer in Westlock, Alberta, using source separated organics from the city of Edmonton waste. And then the Gateway Research Organization is a regional research association that helped us with plot management and field trials. And we also ran these field trials at the Breton plots at the U of A. And so I'm just going to show you a little bit of preliminary data from year one. And so we'll just start with the experimental design and research question. The main question for this project was, what is the impact of source separated organic matter compost compared to synthetic fertilizer on crop yield, fertility, microorganisms, greenhouse gas emissions, and carbon sequestration? And to test this, we use what's called a randomized block design, so which you can see over here. So we had these 10 meter by 4 meter small experimental plots where we randomized these 13 different treatments. We had 13 treatments with factorial combinations of compost, synthetic fertilizer, biochar, wood ash, and gypsum. And in the first year, we grew canola on these, and we looked at and we set these trials up in 3 different soil types. So we set this trial up in an alluviated black chernozem in Wetaskiwin, a solonetic black chernozem in Camrose, and a dark gray Luvasol in Westlock. And so you might have sort of heard Lynn mention that I studied biochar in my PhD, and so biochar is kind of a hobby horse carbon substrate for me. And you've probably heard about biochar, and here's why I think it's an important carbon substrate to put back into prairie ecosystems, and why I think it'll be useful for helping to sequester carbon in agricultural soil. So why biochar? So prairie ecosystems develop with regular fire on the landscape, and research from Saskatchewan shows that up to 65% of the total soil organic carbon content can be black carbon. And so the reason black chernozems are black is because they're full of charcoal. Sorry. So black carbon is produced industrial as a source of energy, which also diverts carbon from municipal solid waste. And so charcoal and biochar are similar on the black carbon continuum. We won't really get into that too much. But we no longer let these ecosystems burn, and we're removing an evolutionary important carbon source. And so because fire has been on the landscape for all time, and therefore black carbon has been in soil for all time, organisms are adapted to it. Organisms have adapted to that regular input of that very specific carbon substrate. And one of the unique things about black carbon is that it has a high capacity for non-polar bonding, which is the kind of bonding that organic matter does. And so essentially what that means is that biochar or charcoal in soil is going to increase the sequestration of other organic matter or other carbon. So we just look at some results really quickly. We'll look at yield. And so there's 3 different sites here. So the blue is the Luvasol from Westlock, the orange is the black chernozem from Wetaskiwin, and the gray is the solonetic black chernozem from Camrose. And these are our different treatments. We'll just start here with the control. So the control treatment was no fertility and no synthetic fertilizer, no compost. This is just sort of raw dog in the soil. And you can see that the yields are not great at any site. And as soon as we add synthetic fertilizer to the black chernozem site, it's got very good yield, 70 bushels per acre. The Westlock site was down around 60, and the Camrose site was down around 55. But as soon as when we added compost, there was a big decrease in the yield on the Westlock and the Camrose site, but not on the Wetaskiwin site. So black chernozem, it was really interesting. Didn't matter if it was compost fertility or synthetic fertilizer fertility, the yield was the same. But that wasn't true in the gray Luvasol or the sodium affected soil in Camrose, which needed synthetic fertilizer. So then you can see in the next treatment here is compost plus synthetic fertilizer. And then again, so the yields go back up. And so now we're actually having, a very similar yield in the black chernozem, but we're having higher yield in in both the Westlock soil and the Camrose soil. And then the next treatment, we get again a decrease in yield. We have compost and biochar, and then compost synthetic. And so we'll just summarize over here. I'll just move this out of the way. So synthetic fertilizer needed on the dark gray Luvasol soil, but then compost, biochar, and synthetic fertilizer had the same yield or better compared to synthetic fertilizer alone. Churnisms were different. Biochar reduced yield in most cases, but trade-offs with carbon storage. This is a question mark. So as I mentioned before, this is preliminary results, and this is ongoing work. As far as soil organic carbon changes, so the whole point of adding compost and biochar is to increase carbon sequestration. That was a bit tougher to measure. And so we see in the control, we have this, very high carbon content in the Wetaskiwin site, around 6.5% carbon, down around 2% in Westlock, and around 5% in Camrose. And synthetic fertilizer didn't do anything to that, but neither did some of the carbon additions. There's a little bit of change in the carbon, but none of these are statistically significantly different. And so synthetic fertilizer and carbon amendments did not increase soil carbon significantly in 1 year, but this is most likely due to the slow accumulation of total carbon, soil carbon, and reflects the need for more accurate lab methods for quantifying changes in soil carbon stability. So I'll sort of say right here, I was surprised at how much variability there was in soil carbon content. I expected the variability sort of in a flat agricultural field in one quarter section to be less variable than it was. And so that's something else that I think we should be looking into. And then finally, I'll just sort of show you the microbial activity. And so again, this is substrate induced respiration. And so so increasing bars indicates more respiration, which basically means more activity of microorganisms. And so all of the carbon additions increase activity. At Westlock is a little bit mixed at Wetaskiwin, but some definite increases here, and sort of all over the place at Camrose. And I found Camrose hard to interpret because of the sodium content, but biochar in the literature, biochar has been shown to stabilize some of the sodium content too. So I think, long story short, from this research, I think that there's lots of that this is at least preliminary evidence to show that using carbon compost in combination with synthetic fertilizer as a fertility management regime will most likely sequester carbon and is certainly increasing soil function. And what I should have said earlier, and I apologize, I forgot to say this, is that the compost plus synthetic fertilizer treatment, so this one right here was 10 tons per hectare of compost and 50% synthetic fertilizer based on crop nutrient requirements. And so this is 100% synthetic fertilizer, and then we reduced that by 50%, which was massive, and we still had similar yields. And so I think this research is quite promising, and I'm sort of proposing to Ardar that we continue to look at this, but they are not fond of the ROI here. They don't think that the ROI is very good, and so we're going to sort of try to move on to a combination of solid compost and liquid compost, or sort of liquid fertilizer, liquid carbon fertilizers. And I'll talk about that in a moment. So I just wanted to briefly mention a new project that we're starting that I'm sort of looking for support from this group of producers, mixed farmers, and Alberta Beef. So coming back to this paradigm shift to add carbon to fertility best management practices, the 6 C's of regenerative agriculture. So number 3 is crop and animal diversity. And so this project is targeting crop and animal diversity and is a combination between my lab at the University of Alberta, Western Beef Development Center, which is Bart Ladner at the Cow and Calf Forage Systems, College of Agriculture and Bioresources at the University of Saskatchewan, and then again collaborating with GROW, the Gateway Research Organization, Hansen and Land Livestock, which is a producer partner in Westlock, and then Case New Holland Industrial is also going to be supporting this project with some custom intercrop seeding equipment. And so the basic research question and experimental design for this project is, what is the impact of intercropping a commodity grain with a forage? And so then again, we'll sort of look at yield, soil health, microorganisms, greenhouse gas emissions, carbon sequestration, livestock health, nutrition, economics, and ROI. And the idea of the main experimental treatment is canola and forage soybeans in alternate 15-inch row spacing. And with the idea that, and then we're going to compare that to just a monoculture of canola. And then the idea is that we can harvest the canola and swath graze animals on the residue and forage soybeans. And this is work that we're going to propose to the Ag Funding Consortium call this year, 2026, which is the LOIs are due April 1st. And so we'd be happy to discuss this more after the presentation. So the last thing I wanted to talk about was just this changing soil carbon theory, because what I would really like to see in the future is more liquid carbon fertilizer and or compost being used in agriculture. And there's already lots and lots of products available on the market, but the way they're being marketed today is inaccurate, in my opinion. And I'm wondering if we can, agree on the need for a cultural shift in the sort of language used to market this material. And so the historic context of carbon theory is alkali extraction and condensation of large macromolecules, where we quantify things like humic acid, fulvic acid, and humin. And the modern framework doesn't look at that at all. It considers a continuum of chemical and physical protection of known biopolymers, which are things like proteins, carbohydrates, fats, and cellulose. And so I just wanted to talk about this for a minute. And I was talking to a colleague the other day, and he said that we may never be able to get away from this language, but I'm hoping to try. So the historic theoretical framework of carbon stabilization is alkali extraction method, which produces fulvic acid, humic acid, and humin due to these ever increasingly concentrated alkaline and then acidic extractions. And so these molecules are, so fulvic acid is alkali soluble, or sorry, humic acid is alkali soluble and fulvic acid is acidic soluble. But we're talking about, using very concentrated bases and very concentrated acids to break apart this organic molecule. And the fact of the matter is that these fractions do not exist outside of the lab. So no organism ever experiences fulvic acid or humic acid. There's no such thing in sort of natural soil decomposition of organic matter as fulvic acid and organic acid. It's 100% completely a relic of this laboratory method. And the reason is because this is easy to do. It's very easy to do, and this does give you the ability. So, when we look at ratios of fulvic acid to humic acid, it does tell you something about the fertility of that organic matter or the easiness of it to decompose, but it's not accurate for a liquid carbon salesman to come to you and say, this is full of humic acid, or this is full of fulvic acid for your soil. It's going to do great things, because that's not true. And what we should really be doing is changing the language and to be more reflective of the modern carbon framework, which I don't know if we should get into this in too much detail. But basically it involves chemical and physical protection. And it explains why there is very old available carbon in soil. And so the idea is that we have, the soil continuum model. So we have plant and animal residues. These are carbohydrates, proteins, lignin, lipid, pyrogenic carbon, so black carbon. And these degrade over time into sort of large biopolymers to small biopolymers to monomers to CO2. And so eventually this chemical material will become CO2. And whether or not it becomes stable in soils is a function of how long does it stay in these large chemically protected complexes, or how quickly does it become protected inside of aggregates or on mineral surfaces. And so mineral surface protection and aggregate protection is physical protection, and the size of the molecule is chemical protection. And so this is the modern framework for carbon stabilization in soils, that's more accurate and has a lot of evidence to support it. And so what does that mean for sort of this sort of language or changing language, right? So I sort of have this modern framework as a changing lexicon. And so number one idea is that scientific and categorical terms. So researchers often use broader categorical categories that account for the extreme heterogeneity of these molecules. So that's true. So characterizing organic matter is extremely complex. So organic chemistry is insanely complex. And I don't even really do that. I touch on it a little bit, but I'd rather try to make it a simplified lexicon that everybody can understand. So humic substances is the formal umbrella for the term humic, fulvic acid, and humin. Whereas it's probably more appropriate to talk about dissolved organic matter. And I don't mind the term humic substances as being a surrogate for dissolved organic matter and solid organic matter, but not as not. But these, I'd like to. I'm trying to move away from these terms, humic acid and fulvic acid. So the other thing is this sort of emerging and advanced terminology. So analytical techniques advance, researchers are moving towards terms that describe molecular structure rather than just solubility. So, but again, this is sort of overly complex for day-to-day use, but they have these super molecular structures. Some modern theories suggest humic substances are not massive polymers, but rather assemblies of smaller molecules held together by non-covalent forces or non-polar bonding. So again, this is what biochar is dominated by, this non-polar bonding. So this is another reason why I think biochar is critically important for in our in our efforts to try to increase soil carbon and therefore soil health. But the other idea here is a simplified nutritional profile. Can we lump these complex carbon molecules into a simplified soil nutritional profile, including fats, proteins, carbohydrates, nutrients, and fiber? I think that would be much more useful for producers and for even sales and marketing of these products, for use in building soil health. And so, like we have this sort of nutritional profile for livestock, right? So we know what the nutritional profile is for livestock, and we know what it is in every different time in its life, whether it's sort of a pregnant cow or a calf or so the question is, and we know what these molecules are. We know what the molecular structure is of these fats, proteins, and carbohydrates. Can we do the same thing for organic matter? And we certainly can from an analytical perspective, modern analytical perspective, can identify those molecules. And then we could put them into a nutritional profile, right? So something less similar to this soil health profile, we could come up with a soil nutritional profile. And having come up with a soil nutritional profile, I think the next question is, can we actually tailor some of these fertility products to different types of production systems? Like this liquid carbon has a nutritional profile for canola and soybean intercrops, or this has a nutritional profile for canola. And this one has a nutritional profile for wheat, and it's going to help promote those growth promoting bacteria that help with yield. And then the last one is sort of commercial and functional alternatives. Can the future marketplace reflect what these organic amendments are actually made of? So compost can be derived from municipal solid waste, or manure can act as a feedstock for liquid extracts. Liquid carbon extract from compost or manure with specific chemical composition, including percent proteins, carbohydrates, fats, minerals, ash. Biologicals or biostimulants, fermentation products with specific plant beneficial microbial blends, and even biopesticides. So again, these are fermentation products with specific antagonistic microbial blends to reduce pathogens and disease. So just to summarize quickly, soil health is a metaphor for representing the function of a living ecosystem. Soil health is quantifiable with standards being developed currently and hopefully available in Alberta in the very near future. Soil carbon is the keystone substrate for soil health, but the language around its application is changing. Regenerative practices must protect, diversify, and input carbon. And we know we can't rob mineral nutrition from the soil. That's why we use synthetic fertilizer and to sort of maintain our yields. But why don't we believe this about carbon too? Why don't we believe that because we're removing carbon in plants and not increasing carbon sequestration over time, as I think the data clearly shows, why don't we believe we should be adding carbon as a fertilizer? And so just a final idea that I like here is just from a partner, an agrology partner in BC. Soil is a commodity like your tractor. With regular maintenance, it will keep running forever, theoretically, right? Theoretically, run your tractor forever with proper maintenance. And so for soil health, carbon is the maintenance. And with that, I thank you, and I will take any questions. Oops. Yeah.
Thanks, Derek, for that. That was really great. I liked your comment that you said at the beginning, without agriculture, or further, like without healthy soil, we'll be naked, hungry, homeless, and thirsty. And we often, I don't know if we just say this to boost our own egos, but we always say that without beef cattle, sheep, bison or goats, that's what we tell our clients anyway. We'd all be naked, hungry, and thirsty.
Yeah,
So
no, I agree. I agree. And I think it's fun. I really like that. I like that slogan as well. I think the faculty moved away from it because I think they probably felt weird about the naked part, but I think it's hilarious. Still, I like it.
yeah, when you think of everything that grows in the soil, yeah, it's definitely very, very important. I'm just going to start with one question. I see there's some that I've gone up in the chat here, but just going to start with another one that came to mind. Can you just expand on the humic and fulvic acid aspect of you saying like we shouldn't be categorizing it the way we are? Like, is it that the products aren't exactly what they're saying they are?
Well,
Um,
no, so yeah, no, that's a great question. So no, it basically it just means that those terms are artifacts of a laboratory digestion practice. And that doesn't happen in the field. And so organisms don't use very concentrated base and concentrated acid to digest organic matter. They use enzymes. And so those terms are basically artifacts of a laboratory method. And the theoretical framework that was developed sort of the wrong way. It was developed based on these artifacts of laboratory extraction. And so the straight up fact of the matter is that there's no, so if somebody says like, so I was recently touring a facility that makes liquid carbon, and I won't say which one, but they said, as part of the finishing process, we add humic acid to this. And I'm like, no, you don't. I didn't say that, but you don't. I mean, so you're, because what you're saying is that I add an extracted material that is in, pH 1 acid, right? There's like 10 molar hydrochloric acid. You're not adding 10 molar hydrochloric acid extract to anything that's living. And so it's it's tricky though, right? Because we're so ingrained in that language, it's hard to get away from it. But it's not accurate. So I guess the accurate thing to talk about is more like dissolved organic matter or solid organic matter. So compost is solid organic matter, and sort of a liquid carbon extract would be dissolved organic matter. And both of those are going to have a whole bunch of sort of functioning or microbial consortium in there as well. They're going to be full of nutrition. They're going to be full of carbon. They're going to be full of microbes, but they're not full of humic or fulvic acid.
so
But they
so
are they are so they
essentially, go
go ahead.
ahead.
Sorry, go ahead.
I was just going to say, so essentially, they're marketing the wrong thing, essentially.
Well, they're marketing sort of an old theoretical framework. They're marketing so yeah, they're marketing something that doesn't exist. So it's complicated, and maybe it's semantics, and maybe I need to get over it, but it's just not accurate. And so I think what would be better, and this sort of occurred to me, I was at a meeting with the Alberta Independent Agrology Consultants recently, and somebody made that analogy about soil just being an inside out rumen. So soil needs all the same, nutritional requirements as cows do. And so it's basically just an inside out rumen. I thought that was a fascinating idea. Because I think it's accurate, but the analogy breaks down in the sense that a cow has sort of fixed genetic material. It's a closed system with fixed genetics, and so you know exactly what its nutritional profile is. But whereas soil is an open system with unlimited genetic potential. So it's really difficult to know exactly what its nutritional profile might be, but I think it's a good analogy that I think we could be marketing those sort of compost and liquid carbon fertilizers as soil nutritional profile. So this liquid extract is 10% protein and 80% carbohydrates and 20% mineral nutrition,
Yeah.
right? And
Yeah.
so then so you can say, it's got all of this food for microorganisms, but it also has this much NPKs for the plants.
So by improving carbon and like carbon in the soil, like what is happening to mineral availability in the soil?
Well, it should be increasing over time, right? So I think that so increasing soil carbon is going to increase soil function by by, increasing the potential for aggregation, increasing water holding capacity, water retention, increasing microbial function, and therefore, increasing the stock of mineral nutrition. But I don't think you'll be able to, well, we'll get away from it completely. Like I said, I think you're always going to have to be using a blend of synthetic fertilizer and carbon fertilizer. And the reason is that you want to make sure you get enough synthetic fertilizer to maximize your yield for any given growing season, but you also want to be feeding your microorganisms, feeding your soil carbon. So I think that's the other concept that I think is worth discussing more is the idea that soil doesn't eat synthetic fertilizer. Soil eats carbon and plants eat synthetic fertilizer. And so if we want healthy functioning soils, we have to rely on them to provide at least some of the mineral nutrition to plants. And then we would be doing better.
Yeah. Thanks for that. I'll just go into some questions that were sent ahead of time. So you have them in the chat there. So if you need to read them as I'm going through them, based on what's currently available for Western Canada data, how do you know if your soil test results are quote unquote normal? I think you may have touched on that a little bit in looking at the three different soil zones just in kind of central Alberta and
Yeah,
how it changed.
I think in that case you would have to have a conversation with your agronomist and ask them what they're seeing in the region. And so, I mean, I think so. One of the projects I'm working on is also trying to assemble data and assemble sort of a web portal, a web-based system that producers could, submit their soil testing data, their soil health test data, and then download a report that would show them in their region exactly where they are. But that doesn't exist exact that way right now. But what you could do, I think, is rely on the experience of your agrologist who, theoretically sees thousands of soil samples every year from the region and could say, yeah, these fields are really fertile and these fields are not really fertile. And I mean, it's going to be probably fairly intuitive too that the low carbon fields are going to be less fertile or the coarse textured fields are going to be less fertile. But as far as saying what if saying something about an optimum level, I think we need better access to data. Like I don't think that anybody could say that for any given region off the top of their head. They would have to access as much regional data as they could.
Yeah, for sure. Um, is there a tool out there right now that based on where you are located, wait, what soil zone you're in, um, that that could tell you what best crops you could grow?
Oh, that's an interesting question. I think there probably is, but I'm not aware of it because I'm not an agrologist. So my training is in sort of theoretical soil science, and everything I've learned about farming is from Colby Hansen, my farming partner in Westlock. And so like I never worked on a farm as a kid, which I regret. But I might not be here today if I did. Um, so I'm not sure if there are. I think there probably are, but it's again, everything that I'm probably all my answers tonight are going to come back to the fact that I would really like to see a very good agricultural database for Alberta. And I've been working on it for the past 5 years, and we're getting there. We're starting to get there, and specifically with this Alberta Independent Agrology Consultants, I'm really grateful to be connected to them because I think that they have a lot of data, and even with BeefSmart lens, like I mean, I think that if farmers want to, I'm trying to build trust relationships here and sort of be if farmers want to share their data, what I am going to do is promise to never share their data with anybody unless it's aggregated up to, the township scale or greater. But what we're trying to develop are tools that where individuals, agrologists and or producers could um, um, access web-based tools to answer these kinds of questions. Yeah.
Yeah, very good. How do ongoing drought conditions impact soil carbon and soil health?
So I think that I so yeah, again, I think that there that soils with higher function and higher carbon are going to be are going to be better buffered against drought. So that's sort of what the all the theoretical framework says, is that the more carbon you have in your soil, the more aggregation, the more water retention, water holding capacity it's going to have, the better your soil is going to be buffered against drought. And so I think that, and I've heard, producers talk about this too, that soil carbon is the armor for your soil against drought conditions. I think that's, severe drought is always going to be problematic, but that's sort of the way that the knowledge base is moving right now, is that more carbon means more soil health, and more soil health means better drought resilience.
Can you comment on a growing, more diverse plant mix, on growing a more diverse plant mix as a way of increasing the carbon content of soil? So diversity in terms of rooting structure, biennials, perennials, C3s and C4s, probably best done the inclusion of ruminant livestock.
Yeah. I think that's the way to go. And that's certainly, like, so I've been working with Colby for about 5 years, and he's been doing a polycrop of corn with other plants for winter grazing. And I think he's noticed in the past 5 years, he's noticed increased soil carbon content in those. And so again, I as a research scientist, I rely heavily on the literature, right? And so the literature says that the more plant diversity you have, the more diversity of carbon you will have, and diversity is always better, right? So more diversity in carbon is going to mean more carbon sequestration and sort of higher function, right? So it's, and one way to think about it is like the gut microflora, right? Like your gut microflora, the more diversity they have in there, the more healthy you are, and that's what microorganisms are in the soil. It's just the soil's version of a gut microflora, right? And so the microbes are the stomach of the soil, and higher plant mixes means higher carbon, it means higher below ground carbon deposition from roots that is different, or higher diversity, and therefore more functioning microorganisms. And so, yeah, so a combination. So that's what Colby's, yeah, combination of C3 and C4 plants. So Colby's on the call here. He can chime in if he wants, I guess. But there's like corn, sunflowers, um, some peas, different end fixers, and I think, yeah, like I said, he's noticed a difference in carbon sequestration over the past 5 years in that stand. But then, yeah, so he leaves it standing. So he grows this polycrop and leaves it standing as winter forage for cows, which I was fascinated by. I asked him, I'm like, do they eat everything? He's like, yeah, they start with the ears of corn and they eat everything. They eat it right to the ground, everything. I'm like, that's cool.
It's very good. One more question. How does the level of organic matter in the soil relate to retaining or improving carbon levels?
So soil organic matter is 58% carbon. And so they're directly related. And so soil organic matter just includes all of the other elements in organic matter. So when you say, so when you say soil organic matter, you're including carbon, hydrogen, oxygen, nitrogen, all of these sort of macro and micro elements. And so soil organic carbon is just elemental carbon. And as a rough estimate, soil organic carbon is 58% of soil organic matter. And so if you have soil organic matter in one year from your agrologist, and you have soil organic carbon another year from maybe a different agrologist or a different lab, you can compare those numbers. You can do some calculations to see if it's been going up or down. But
Oh, thank you. Thanks for
yeah.
that. Do you want to expand on your research that you're going into by including livestock?
Oh, yeah. So yeah, that's a project that I'm talking to a research representative from Alberta Beef this week. And so Bart Lardner and I and Colby have been talking about this project for a while, and he's got Case New Holland interested in designing some equipment to make it easier to do this. And the idea, yeah, is that you by having sort of a couple of different species of plants, again, you're increasing carbon diversity below ground, you're going to increase microbial function and theoretically increase carbon sequestration over time. That's a good thing. But then you get a sort of a commodity crop yield, right? So you harvest canola, and then the residue of that is still has, enough protein and carbohydrate content that and the foraged soybeans in the intercrop, you swath graze all of that stuff. So then you put cows on it for some number of grazing days as well. And so we're going to test that. But what I was hoping to get an idea. So one thing that Ardar and the Ag Funding Consortium always ask questions about: what is the likelihood of uptake for this practice? And I asked that to you one time a couple weeks ago, Lynn, and you said that you thought it was really high. And so I guess I was just curious if the people on this webinar, or um, people that might be watching it later on, if they wanted to email me, um, if they think the likelihood of that is high, or if they would uptake that, right? And so what I've been doing is I'm trying to sort of build up a network of partners through organizations like BeefSmart and the Alberta Independent Agrology Consultants. And I send a Google form out that sort of goes as a memorandum of understanding about how I will use and protect your data. And so maybe that's something that I can send you. I'll send you my business card, and maybe you can like freeze, like photo capture it into the video at the end or something. Because I'm always, like I'm trying to build up this network. And so I guess that's the question is, yeah, do folks on the call think that there'd be uptake for that, for more of that sort of commodity crop than slash swath grazing afterwards? And so the research is really just trying to quantify that, trying to put numbers to it, sort of trying to look at the difference in livestock health between monoculture swath grazing and this sort of polycrop or this intercropping swath grazing, and then the soil health metrics, to all the soil health metrics and some of the microbial metrics.
Yeah, no, that's very good. Thanks for that summary. We'll definitely add your contact information, and we could even add your Google form for questionnaire at the end,
Yeah,
and
yeah, I'll
pass
send
that
that to
on to
you.
everybody.
Yeah, that sounds really good. And because the other thing is that I'm also working with the Regenerative Agriculture Lab, which is part of Climate Solutions Lab. Climate Solutions anyway, and they're running a polycropping trials too. And so looking for farmers um, to collaborate on an intercropping trial as well.
Well, you're in luck because we have a lot of clients
Yeah.
that do intercropping and different types of blends, from fairly simple 2 or 3 species blends and versus to 16 species. So, and yeah, definitely swath grazing or post harvest grazing is definitely included. And if we can, I know it's been a little over an hour. There's one more question that popped up. Um, let's just take this question, guys. I know this is a common question that we get. Can
Mhm.
you touch on the physical impact of soil health of using cattle to graze residue? So, like
Yeah.
the
So
the
everything that I've read about that is that it still has a positive effect because you're grazing cattle on the site. They're leaving all of that organic matter on the site, right? So Bart was saying that cattle are only something like 80% efficient at taking up nitrogen, and or 20% efficient. So they excrete 80% of the nitrogen
80%.
and 90% of the phosphorus. So all of that stays in the soil, which is a good thing. And all of the carbon, all the non-digestible fiber and carbon stays in the soil. And so you're improving carbon content, and by improving carbon content, you're increasing aggregation. So you're having a positive effect on the physical properties of soil. Um, And so really, I mean, that's why I sort of said at the end there in the summary that carbon is the sort of is the keystone substrate for soil health. Sort of carbon, and it sounds like sort of a one-stop shop, but it's going to be harder to implement, I think. But it is in a sense. It's carbon is the number one thing you can do to improve soil health and no-till.
No, that's very good. Thank you for that. So we with that, just to not go too long, I know we could ask questions, Derek, for another hour. I'm sure this is very interesting what you've brought up, and you brought us a lot of information. But I hope everyone you enjoyed our discussion tonight. and had a few key takeaways to consider before you begin your seeding practices this spring. Perhaps you can tweak your practices based on the information that was shared tonight. Please reach out to Derek or I if you want to discuss any of the topics or need any other information. I know Derek's very quick at getting back information and always open to discussing these matters further. So a link of the recording will be sent out in a few days, and I encourage you to share it with anyone who might find it interesting. And of course, we're always open to feedback. So if you have any other topics you would like to see covered in our future webinars, please don't hesitate to let us know. Keep an eye out for more invitations for our future webinars. Our plan is to have another one in a couple of weeks as we're continually planning to share our knowledge with all of you. With that, I'll end the recording and thank you all again for attending tonight.
Great. Thank you, Lynn. Thanks everyone for joining. And I just reiterate, I'm always available for phone call or email chat. I tell my students I'm always on campus every day and never very far from a computer these days. So thanks again.
Thanks again, Derek, and have a good night, everybody.
This presentation has been brought to you by BeefSmart Consulting Inc.